The reason why we call it acellular bacteria because this refers to bacteria without cell wall, because these are not just your ordinary bacteria. These microorganisms lack peptidoglycan. So that's the reason why we call them acellular bacteria. Okay? So the first group that we will be discussing, so we call it the PPLO. PPLO, which stands for PPLO stands for pluro pneumonia like organisms. Okay? PPLO are significant human pathogens, and they belong to the class molecules. Okay? These class molecules include the following. We have the Mycoplasma, Ureaplasma, Acoleplasma, Spiroplasma, Thermoplasma, and Neuroplasma. But we shall only be concentrating our discussion with Mycoplasma and Ureaplasma. Okay, so the PPLO, these organisms are the smallest self-replicating organisms. These organisms lack cell wall. So they lack cell wall, and they are only enclosed by cytoplasmic membrane. So the two important genera of Mycoplasma includes the Mycoplasma, and there are about 69 species, and the other one would be the Ureaplasma. There are 2 species, but we shall only be concentrating on Neuroplasma urealyticum, Mycoplasma pneumoniae, and Mycoplasma hominis. Why do we call it pluro-pneumonia-like organisms? Because of the fact that they don't have any cell wall, they don't have any specific shape. Therefore, we cannot classify them as cocci or bacilli. The shape of the organism depends on where they are attached to. So later on, I will show you the morphology. So in summary, as you can see, they're so small, about 0.2 to 0.8 microns. In fact, the colonies are also so small that you have to use a stereo microscope for you to visualize some of the colonies. Okay? And the cell wall is not present, so you don't expect to see peptidoglycan. And instead of peptidoglycan, they have sterols. And sterols are also needed for them to be grown. They are facultative anaerobe, divide by binary fission in 1 to 6 hours. They are resistant to penicillin and cephalosporin. Why? Because the action of penicillin is that it destroys cell wall. Wala naman silang cell wall, hindi destroy sa kanila. Di ba? So, effective antibiotics would include tetracycline and erythromycin. So, since they do not have cell wall, sometimes we call them CWDs, or the cell wall deficients. They're slow-growing and fastidious. They would require cholesterol and fatty acid for growth because of the sterol. And then, so 2 Mycoplasma species are common to humans, and these are the Mycoplasma pneumoniae. If you are familiar with primary atypical pneumonia or the walking pneumonia, okay, that is caused by Mycoplasma pneumoniae. And the Mycoplasma hominis is considered as the smallest free-living organisms in nature. Quite difficult to detect. Common contaminant of cell culture. Okay? And they are considered as emerging pathogens. Mycoplasma, like what I've told you before, they are known as the PPLO, or the pluro-pneumonia-like organisms. If you will be looking at the colonies, they appear like fried egg appearance, and first isolated in bovines, these are in cattle, as pleuropneumonia. That's the reason why it is known as the Eton agent, because the one who discovers it has a surname of Eaton. Eaton? Eaton? Okay. Okay. So, that is discoverer. Okay, the Eaton agent. Okay, so where is the location of infection? It will infect the epithelium of the mucosal surfaces, particularly in the respiratory and urogenital tract. They will adhere tightly to the epithelial cells. Why? Because they don't have a shape. Diba sabi ko kanina, their shape depends on where they are attached to. And they can also infect the oropharynx, particularly for Mycoplasma pneumoniae. But for urogenital tract, mas common yung Mycoplasma hominis, particularly among women. Okay. So, this is the fried egg appearance of Mycoplasma. So, this is how the colonies would look like. Okay, so the fried egg appearance colony. And how do you think they are transmitted? Mycoplasma pneumoniae could be transmitted by droplets. Whereas Mycoplasma hominis and Ureaplasma ureaticum could be transmitted by kissing or by sexual intercourse. So they are found in urogenital tract. Causative agent of non-gonococcal urethritis. This is the Mycoplasma pneumoniae. This is the scanning electron micrograph. Can you determine what shape it is? No. No particular shape. Because they don't have peptidoglycan, they don't have a cell wall. So another electron micrograph. So if you cannot decipher, this is the cilia of the trachea. This is the cilia of the trachea, and they are attached here. Okay, so there's no particular shape. So this is another example of electron micrograph. The Mycoplasma is here. Can you see the MV? MB, this refers to the microvilli. MB refers to the microvilli. The letter C here, that's the cilia. And somewhere in between the microvilli and the cilia, you have here the Mycoplasma. So the shape depends on how they will fit into your trachea. Can you imagine what I'm trying to say? So, the diseases are bronchitis, pharyngitis, and the walking pneumonia, known as the primary atypical pneumonia. Mostly, bakit siya tinawag na walking pneumonia? Di ba most of the time, when you say, he has a pneumonia, ano yung unang mapapasok sa isip nyo pag may pneumonia yung tao? Bedridden, nahihirapang huminga. Di ba? Ganun yung mga tao may pneumonia. But itong mga taong to, kahit na meron silang pneumonia, they are asymptomatic. They are literally capable of walking. Kaya siya tinawag na walking pneumonia. Isolation always indicates pathogenicity. You cannot say, it's a normal flora. Okay, so 20% found in school-age children and young adults, and even 50% in military settings. So, they are spread via close contacts such as droplets. The incubation period would be between 2 to 3 weeks. And signs and symptoms include headache, fever, malaise, anorexia, sore throat, dry cough, and even earache. So, as you can see, this is an illustration, a much better illustration. So, this is the epithelial cells. And this is the organisms. So, ito yung sinasabi ko sa inyo, that the shape of Mycoplasma depends on where they are attached to. Sila yung nag-a-adjust. Kung ano yung, ito na lang yung kasya na space. So, this is how they would adjust to space. Can you now imagine? So, that's why they are known as the pluro-pneumonia-like organism, the Mycoplasma. So, this chart here will tell us the duration of the Mycoplasma pneumoniae. Can you just imagine? You still be positive for antibodies for Mycoplasma pneumoniae for at least 40 days. And you know what's so special about the antibodies of Mycoplasma pneumoniae, particularly the IgM antibodies? The antibodies are also known as the cold agglutinins. Cold agglutinins. Anong ibig sabihin ng cold agglutinins? Cold agglutinins means that the antibodies will agglutinate when incubated at 4 degrees centigrade. They will agglutinate red cells when incubated at 4 degrees centigrade. So, you'll have fever, particularly from 5th until the 10th day, but bumababa ang fever when the days become as days go on. Okay? Headache usually increase optimally between 10th to 15th day. Chest soreness from 5 to 10 days. And then headache and malaise from 10 to 15 days. Dry cough from 10 to 15 days. Okay, so this is how the organisms, the infection would progress. Okay, clinical manifestations of primary atypical pneumonia. So, primary signs and symptoms would include headache, fever, sore throat, cough. Okay, and then signs will be you have to collect the sputum. Okay, and dullness, and patients would also be characterized by dullness and rales. So that's it for Mycoplasma pneumoniae. But there's another Mycoplasma, okay, that infects the genitourinary tract. So this includes the Mycoplasma hominis and then the Ureaplasma. Ureaplasma urealyticum. As you can see, Mycoplasma hominis can cause in male urethritis and prostatitis. Ureaplasma urealyticum could cause vaginitis, kidney inflammation, pelvic inflammatory disease, postpartum fever, and spontaneous abortion. So, Mycoplasma hominis can cause the following infection. Infection of the urogenitary tract. Do you know that Mycoplasma hominis is normally present for some of us, particularly in our urogenital tract. However, they can cause upper urinary tract infection. When you say upper urinary tract infection, so this may go as far as your ureter, to your bladder, to your kidneys, not just to your urethra. Particularly for those people who are sexually active. Kaway-kaway. Okay, so these organisms are considered as opportunistic pathogens, and they are capable of causing infection to immunocompromised individuals. When I say upper respiratory tract, upper urinary tract, napaisip din ako din ng respiratory. Upper urinary tract, nakaabot doon, no? Okay. Upper urinary tract, this will include inflammation of the fallopian tubes, particularly for female. We call it the salpingitis. Inflammation of the kidneys. We call it pyelonephritis. And it's dangerous for female because there's a chance that you might have PID or pelvic inflammatory disease. You know what will happen if you will have that, right? So you'll have a very difficult pregnancy, or sometimes you'll not be able to bear child anymore because of the pelvic inflammatory disease. PPLO, another one is Ureaplasma urealyticum, causing non-gonococcal urethritis. Again, if Mycoplasma would infect upper urinary tract, Ureaplasma will infect lower urinary tract, but can still infect upper respiratory tract. It is significant due to infection of the fetus. particularly if the female is pregnant. Siyempre, wala naman kayong fetus if you're not pregnant. Okay, so, it will cause chorioamnionitis or the infection of the placenta membrane. So, significant to. Kasi the child could be affected. And then, congenital pneumonia. And then, chronic lung disease in premature infants and meningitis. Imagine, the infection is actually coming from the mother, but because the organism goes to the upper genitourinary tract, so it would affect even the fetus. So this one is very significant for pregnant women. Okay? Aside from women, it can also infect men. Particularly, it's the etiology of non-gonococcal urethritis in men. What do we mean by non-gonococcal urethritis? The signs and symptoms are similar to gonorrhea, except it is not caused by gonorrhea. It is caused by ureaplasma. So that's why we call it non-gonococcal urethritis. Other species would include Mycoplasma genitalium. This is also associated with pelvic inflammatory disease and non-gonococcal urethritis. And there's a question about AIDS-related mycoplasma. Okay, the AIDS-related mycoplasma, we call it the Mycoplasma fermentans. So it might be asked every now and then to you. So what's the AIDS-related mycoplasma? It's Mycoplasma fermentans. Okay, so aside from them, we also have the salivarium, orale, buccale, photium, lipophyllum, fermentans. Okay, so, but of course, yung pinaka-common na na-discuss natin, these were Mycoplasma pneumoniae, Mycoplasma hominis, and Ureaplasma urealyticum. So, how do we collect specimen? The organisms are extremely sensitive from drying since wala silang cell wall. So, we have to use a transport medium, and the transport medium usually is the TSB. Then we have to add about 0.5% albumin, and we also have to add penicillin to remove normal flora. Anyway, they are not affected by the presence of penicillin. The proper pronunciation is penicillin. Okay? If not plated, we have to immediately freeze the specimen because they are actually sensitive to drying. For Mycoplasma pneumoniae, we generally do not culture because it will be so long and the sensitivity is very low. Instead, we have serological tests, such as we look for the presence of cold agglutinins. Ano nga yung cold agglutinins? IgM antibodies that can cause agglutination of red cells when incubated at 4 degrees centigrade. For Mycoplasma hominis and urealyticum, we can culture them in liquid media, and we watch for pH change. Okay? So, and then, we plate them in enriched culture medium. When you say enriched, the culture medium should have sterols. Sterols are needed for the growth of Mycoplasma. For Mycoplasma hominis, the enrichment is arginine broth. What do I mean again when I say enrichment? These are liquid or solid? Liquid. Before transferring it to solid medium. Nasa tube muna bago Petri dish. For 7 to 14 days. The enrichment broth for Mycoplasma hominis, we call it arginine broth. So if after 14 days, from salmon light color, naging pink na siya, it means the arginine has already been released. So we can now transfer it to the agar. In the agar, we have to look for the presence of the fried egg colonies appearance. And we usually stain the colonies with the dye stain. So the dye stain, kasi class, ang colonies ng Mycoplasma hominis, sobrang liit nito. Di ba pag tinignan natin yung colonies ng Klebsiella, makonki, kitang-kita agad natin, di ba? Ito hindi. hindi nyo agad makikita. Kailangan nyo pa mag-stereo microscope para makita niyo yung colonies. Tapos, kailangan nyo pang stain. Hindi yung bacteria, ha? I-stain niyo yung colonies mismo. Ano yung pang stain? Dye stain. Imagine niyo yung egg yolk. Egg yolk. Okay. Yung yolk mismo, dark blue. Tapos, yung pinaka-egg white, light blue. Parang ganito yung itsura ng colonies ng Mycoplasma. Ureaplasma, instead of using arginine broth, ang gagamitin naman natin dito, U9B broth. Miss Enahe, U9B broth. Okay? So, in 3 to 5 days. Kasi kanina sa arginine, 7 to 14 days. Ito lang, ang gagamitin natin, 3 to 5 days. So, yung broth, hihintayin nyo mag-pink. Kasi originally, yellow yung color ng broth. Pag naging pink na siya, it means ammonia has already been released. Tapos, ipa-plate na natin siya sa agar. Anong pangalan ng agar? A7B agar. Tapos, irregular yung shape ng colonies, we usually add magnesium chloride. Okay? So, pag nag-add tayo ng magnesium chloride, you would expect the colonies to become rusty brown. Kasi, it has already been converted to magnesium oxide. So, titingnan ulit natin sa stereo microscope. Kasi maliliit lang yung colonies nito. Okay? So, ito, sinasabi ko. Ito yung itsura niya ng PPLO. Anong shape? Bacilli? Cocci? Wala. Di ba? Wala siyang shape since wala siyang cell wall. So, anong tawag dito? Pleomorphic. Pleomorphic. They can assume a variety of shape. Ito yung sinasabi ko kanina. Yung colonies ng Mycoplasma hominis. Tingnan nyo to. Etong buong to, tingnan nyo, imagine nyo kung gaano kaliit ha. Etong buong to, ito yung colony ng Neisseria gonorrhoeae. Ito yung colony ng Mycoplasma hominis. Ganun siya kaliit. So, kaya kailangan nyo talaga mag-microscope. Stain, tapos mag-microscope. Ito yung culture ng Mycoplasma hominis. Ito, rusty brown color. Naalala niyo sinabi ko kanina? Upon addition of magnesium chloride sa agar, mag-rusty brown color. Ureaplasma rhealyticum. Ito yung kulay blue, Mycoplasma Pneumoniae. Dye stain. Ito yung dye stain for Mycoplasma. 'Di ba? Dark blue yung egg yolk, 'di ba? Fried egg appearance. Tapos light blue yung parang egg white na, light blue yung color. Dye stain to for Mycoplasma. The only way for you to see the colonies of Mycoplasma. Ito, black and white illustration of mixed sizes of Mycoplasma. When stained using DNA fluorochrome stain. Okay, so we have here different laboratory methods for detecting Mycoplasma and Ureaplasma, divided into two main types, non-serologic and serologic. Ang tandaan nyo lang, kapag non-serologic, this includes culture, indirect immunofluorescent, or PCR. Pag non-serologic, pwedeng complement fixation, immunofluorescent antibody, latex agglutination, or enzyme immunoassay. Anong ibig sabihin ng serologic? We look for the presence of antibodies galing sa pasyente. Anong ibig sabihin ng non-serologic? Pag walang antibody testing, non-serologic yon. Pwede nating i-culture, or direct exam, or we amplify the DNA. Kapag sinabi nating amplify the DNA, ano yon? PCR. Okay, so ito, Ureaplasma rhealyticum. Ito tingnan niyo ha, kung gaano kaliit. Kung maliit na si Mycoplasma, mas maliit si Ureaplasma. Okay? Alam nyo kung anong other name ng Ureaplasma? T-strain. T for tiny. Ayan, T for tiny. Gandang nickname yun, tiny. Tiny. Ayan, di ba? Treatment. For Mycoplasma hominis, the treatment, if you are resistant to erythromycin, then lincomycin could be used. For Neuroplasma erythicum, if resistant to lincomycin, then erythromycin could be used. Parang niloloko tayo ng slides na to, no? Okay. Mycoplasma pneumoniae, we can use erythromycin and tetracycline.